This is our osmosis and diffusion through a membrane lab. And we started off by making a hypothesis. And the hypothesis for the glucose was that if cellulose tubing full of a glucose solution was placed into distilled water, the glucose would diffuse out of the cellulose membrane and into the distilled water. And our hypothesis for starch was that if a cellulose tubing with starch solution was placed into distilled water and the iodine was added to the water, then the starch would diffuse out of the cellulose tubing and into the water. So we started off our procedure. We added the glucose solution into the cellulose tubing, and then we placed it into 250 milliliters of distilled water, and we took the pH, and then for the starch we placed the starch solution into the cellulose tubing and then placed that into 250 milliliters of distilled water and then added iodine. We found that the results were that for the starch, the iodine diffused into the cellulose tubing, and so we were wrong because we thought that the starch would diffuse, but the iodine diffused into the cellulose tubing. And then for this we were right. The glucose did diffuse out of the cellulose tubing and into the solution, so the two solutions became isotonic.
In conclusion, we can say that the water and iodine diffused through the dialysis tubing, and the starch did not, because starch molecules are too big to fit through the pores in the dialysis tube, in the cellulose dialysis tubing, whereas the water and iodine molecules are small enough to fit through, and glucose molecules, like the lab says, are considerably larger than the water and iodine molecules, but they're still small enough to diffuse through the cellulose membrane. So that's why we had the results we did. And I guess from the lab we can assume that the membrane, its pores are of a specific size, designed to let in sizes of certain molecules, whereas they're small enough to not let others in. So in other words, it's selective.